Hi, I'm Richard Sever from Cold Spring Harbor Lab and BioArchive. With me, I have Olivier Pourquié from Harvard, who is here virtually at the Cold Spring Harbor Symposium on biological timekeeping. Welcome, Olivier. We were just saying it's a shame that you can't be here in person, but Zoom is the next best thing these days, I think. So you gave a fascinating talk the other day about developmental timing. Can you just kind of give us a bit of a high-level view of the differences in timing that we see during development? Because what was interesting to me was you showed a kind of picture of a mouse embryo and I thought it was a chick embryo and a human embryo that were kind of like, you know, very different timescales, but the embryo looked very similar, and it was almost like these are, you know, it reminded me of when I was a kid being told that, you know, I'm 24 in dog years or something like that. So can you say a little bit about this difference in timing in embryonic development?
Yes, sure. So it's, I mean, it's very striking if you look across evolution that there are tremendous variation in the timing of development. So there are two aspects. There's, you know, one way to think about it is if you think about cold-blooded animals, for instance, is temperature. So it's well known that temperature will control the pace of development. And so the meeting was a lot about circadian clocks, which are temperature compensated. But here in development is not temperature compensated, except in homeo terms. But say, for instance, if you take a frog embryo and you progressively increase the temperature, it's going to develop faster. And that's true for a fish embryo, for you know, all these cold-blooded animals and invertebrates as well. But now if you take amniotes like birds, for instance, or homeo terms, say the birds or the mammals, what is very striking is that they develop at 37, but still you see these dramatic differences in the developmental timing. So for instance, the chicken embryo I showed was 4 days, and it's morphologically similar to a 6-week human embryo. So it's tremendous difference in the timing. And so I've been interested in this for a long time, and so I did my PhD in France with Nicole Ledoirin, who developed the quail-chick camera system. And so we were playing always with these different species. And one of the recurrent themes was the difference in timing, of course, which is quite subtle between quail and chicken, but still very significant. And then I became interested in somatogenesis, and that is the formation of the precursors of the vertebrae. And one of the very striking aspects of this system is that they form in a periodic fashion. So the precursor of the vertebrae are added one after the other by pairs in the embryo. And this their production obeys a defined rhythm. And this rhythm is characteristic of the species. And there are very significant variations, like being 30 minutes in zebrafish and 90 minutes in chicken, two hours in mouse. And so that's, I've always felt that this was a great handle into this question of developmental timing, except that for a long time, the tools were not right.
Right. So yeah, I mean, one of the phrases that you used in your talk was that they're a kind of a ruler, that somites are a ruler for developmental timing. So across, I mean, so you mentioned these are kind of like what basically an early stage of the different segments of the embryo along the spine. So that's really the case, is that you can plot all vertebrates using this ruler?
It's a bit more, so within a species, it's definitely a ruler. So that's within a species and within a developmental period, which is somatogenesis. So when the somites are forming, which covers a significant period of early development, and which is also an interesting period because it's what people call the phylotypic stage. That's a stage where vertebrate embryos are very similar, or at least that are the most similar. And then within this phase, and that's, you know, that's what embryologists have done for ages, is really to count somites in order to compare your embryos, because otherwise it's very hard to know that two embryos are at the same stage if you don't have a real measure. And morphology is very difficult because, you know, the limb bud can be a little bigger, a little smaller. It's hard to be precise, but somite is it's integer. I mean, you count, it's a 15 somite embryo or it's a 20 somite embryo. It's precise within the window to make a somite.
Right, I see. I mean, yeah, because you showed a couple of great videos where, you know, you have these kind of new stripes that appear with this kind of regular periodicity. So what's actually happening as one of these stripes forms? What's happening sort of at the sort of developmental cell level?
Mm-hmm. So the idea is that when a somite form, so the vertebral column forms in a head to tail fashion. So as I said, new pairs of somites are rhythmically added until you reach the tail. and every time a new pair of somites is formed, there is a wave of gene activation that traverses or that sweeps across the precursors of the somite. And so these waves of transcription they occur in response to periodic signaling that is downstream of well-known signaling pathways like Notch, Wnt, and FGF. And they're periodic. And so that's why we proposed that this was identifying a molecular oscillator that we termed the segmentation clock. And so the idea is that this oscillator controls the rhythm of somite production.
Right.
But there
where does a wave start? I mean, that was another thing that you showed this. I mean, you did talk about a wave up and down, but you saw this great sort of GFP wave was, I think, in the embryo.
So the wave starts in the tail bud, that's the posterior extremity of the embryo, and then it moves up and it stops where the future somite is going to form. And in fact, so the clock, so the molecular oscillator that produces the waves, is one of the ingredients of the system. But then there is a second one, which is called the wave front, which is a posterior gradient of signaling, which involves Wnt and FGF, that defines a competence window. So it basically gates the response to the wave, to the clock, and such that only the cells that passed this particular wave front during the previous cycle, only these cells can respond to the clock.
Right. So I mean that is all obviously happening in a whole developing embryo. But you talked about the fact that you've managed to set up an in vitro system to allow you to find out what's going on. Can you tell us a little bit more about how that works?
Yeah. So I mean, obviously, this is a very complex behavior with this system. So there is an elongating embryo, oscillations, and there is a spatial temporal component, which is which makes the system hard to challenge and to manipulate. And so, after the discoveries of stem cells and Yamanaka reprogramming and all that. I decided to try to see if we could simplify the system and recapitulate this behavior in a dish using embryonic stem cells. So we started with mouse, and then we moved to humans, and eventually we and also the group of Kanta Salef in Japan and Kageyama were able to recapitulate this behavior in the dish that is to differentiate these human cells to a presomatic mesoderm stage, so the stage where you see these oscillations of the clock, and to show using reporter that once the cells reach this stage, they recapitulate the oscillations. Right. And then we have something we can measure, like we can estimate the period, and we can generate loads of cells, and they are synchronized, and it's very, I mean, I wouldn't say easy, but it's much easier system to manipulate.
So this is what, just sending them back to iPS cells with one set of factors and then sending them just as far as the pre-Semitic mesoderm with another set. Is that how it works?
Yeah, exactly. And it's amazing because with, in fact, from the iPS, so from the pluripotent stem cells, we only use two compounds, two small molecules, and it gives us almost 100% of human pre-Semitic mesoderm cells, which is a tissue which is impossible to get because in humans, this tissue is formed between the 3rd and the 4th week of gestation, which is a stage at which women don't even know they're pregnant. So there's almost no data on this stage.
So, and these, I mean, it's amazing to me because what you have described in the whole embryo is there's this process that has these temporal and these spatial effects, and then you're basically, you know, recapitulating this in cell culture. How much can you recapitulate? You get the same kind of, can you create this periodicity that you were seeing in embryos?
Yeah, absolutely. So you can get the periodicity. You can, it's absolutely amazing. So my group and also Kantas Aleph, who's been working in Kyoto University on the same. So we both developed systems now where we can make actual somites in vitro, and we can make them either in a sequential fashion or in simultaneous fashion. I mean, the possibilities offered by these systems are just mind-blowing. And you can control, so you can make only the tissue of interest, which in this case is the tissue that forms the somite, nothing else, which tells you, you know, I've been working a lot on signaling and the influence of the different tissues on each other during development. There's no other tissue. It's pure paraxial mesoderm, which develops and gets pattern in total autonomy, which is mind-blowing.
So from a kind of, from a sort of signaling perspective, what are you putting in? Are you putting in things like wingless, FGF, notch, that type of thing?
We just put the only key ingredient to trigger the whole stuff is a Wnt activator, which is in fact an inhibitor of GSK3 beta called Chiron. It's a small molecule. And we also add an inhibitor of BMP, because BMP will tend to inhibit the pre-Semitic fate, so it will make things more like clean buds also.
So that's the non-developmental biologist, that's a TGF beta family.
Yes.
Right. Yeah. And what's your readout? I mean, you showed things flashing green. What's your readout for the peak of these waves in the in vitro system?
So that's in fact what we read. So we introduced fluorescent reporter, which is it's a optimized version of GFP, which is very destabilized because if you want to see the oscillations, you need an unstable protein. And this construct is introduced into the locus of a gene which is regulated by the clock.
Right.
So we know that there are periodic notch oscillations, so it's a notch target, basically.
Right.
And so what you see are notch oscillations in this system.
It's a readout of notch oscillations. And one of the things you mentioned was that you see these correlations with what, body mass, lifespan, and metabolic rate. These are all things that you can correlate with these oscillations. Is that correct?
Yeah, absolutely. That which was a surprise, because I mean, if you look at this literature on correlations between metabolic rates and mass and so on, it's been well worked out. There's this Kleiber law that dates back from 50 years ago that says that the metabolic rate of an animal scales with the mass, but with an exponent of 3/4, which means that it's not as so there's not a linear correlation between the metabolic rate and the mass. So the larger the animal, I mean, as the animals grow larger, their metabolic rate increases, but not exactly in proportion. it's a bit slower than the increase in mass. But anyhow, the point is that this was proposed to be caused by the delivery of the energy, so the organization of the blood supply. And so there've been a lot of theoretical papers produced on that. And it was never really argued to be coming at the cellular level.
Mm-hmm.
So there's been some discussions about that, but they've been kind of ruled out by these predominant models. And so what was really surprising was to see that in the embryo, at least in this tissue, and we also have evidence for another tissue now, there is definitely a correlation between the metabolic rate and the developmental timing. It's not such a surprise, I would say. I mean, it's very intuitive, that if you do things much faster, you consume more oxygen, more energy, basically.
Right. So, yes. I remember hearing that there are rodents, for example, that are of similar sizes, but have very different lifespans. Is that something that you're looking at to tease out the details?
I would like to. I mean, the one would be like the naked mole rat. It's not really easy to access this region, so I don't know how much we'll be able to. But now there are more and more iPS cells from OES cells from rat, from various mammals, from bovine, from monkeys, and so it should be quite reasonable to have access to at least a few selected species along.
Anyone you can get ESLs from, then you can do it. Yeah.
Exactly.
I mean, one of the things that was, you mentioned this notion that there was this sort of blood supply physiological level that was one argument, and then you look at this, the readouts, and it's a fundamental correlation with metabolic rate. I was kind of intrigued to see you showing that suddenly you're putting in electron transport chain inhibitors. Can you explain what was going on there?
Well, so what we see when I talk about metabolic rate, it's measured largely by oxygen consumption. So, and oxygen consumption means basically respiration, which is the place where most of the oxygen is consumed in the cell. And so that really leads us to the electron transport chain, which is where the oxygen is used. And so we decided to try to see what happens when we inhibit the electron transport chain. And then we saw that it does slow down the period of the oscillations. And so if you consider that the period of the oscillations of the clock is a readout for the developmental timing, that means that by slowing the respiration, you slow the developmental timing, which would be consistent with this higher metabolic rate driving the faster development. What was surprising is that we see that when we block the electron transport chain, but not the ATPase, because the goal of this chain is to really remove electrons from NADH, which is produced by the Krebs cycle, that essentially down from the glucose consumption. You remove electrons, and then through a series of transport, the energy of these electrons is used to pump protons out of the mitochondria into the intermembrane space and generate this proton gradient, which is used to make the ATP. But what we see is that if we block the production of ATP, or if we disrupt the proton gradient, we don't impact the period of the oscillations. It's only when you act on the transport of the transporters of the electrons. And this is not so easy to explain. What is this? Why you get this?
So it's not about energy or ATP per se. Then it's about complex 1 to 3.
Yeah, exactly. It's more redox or NAD. It might be that you really need to regenerate the NAD because for many, it's important for many reactions in the cells, and so maybe that might be the limiting factor.
So do you think it's a correlation? But I mean, if I remember rightly, so that's you've got to do a whole bunch of biosynthesis. And that's what you've got to build a whole organism, and it's more about having the capacity to build the organism rather than the energy to do so, perhaps.
Yeah, that's what it seems. Yeah, because ATP, I mean, we have more arguments which I didn't have time to show. It doesn't seem to be. If you increase the ATP production in the cell, it doesn't speed up the clock.
Right. Well, it's fascinating because, I mean, in some respects, it reminds me of some of the discussions that I've known have been going on about cancer biology and the Warburg effect, when people suddenly, there was this point, well, why is the cancer cell depending on glycolysis? And it's, well, because it's, it's more about diversion of carbon skeletons for synthesis than it is about creating ATP in the Krebs cycle. There's some, it feels like there's an echo there. So that's fascinating.
But that's interesting, because actually we've published several papers showing that these cells that show the oscillation exhibit this Warburg effect. So they show this very active glucose consumption, which is mostly converted to lactate. And they also show an inverted pH gradient, which is characteristic of cancer cells. So there's a lot of similarities between these cells and cancer cells. Actually,
well, that's fascinating, because there's so many things that sort of show up in cancer, which people think are unique to cancer cells, and then you realize that it's recapitulating something that happens in development.
Yeah.
I guess with all these things, that means you suddenly got a whole new wide spectrum of potential collaborators.
That's right. Yeah.
So what's the next step then?
So next step would be really to figure out what's happening. I mean, there are two important things we need to figure out. One is, why the electron transport chain, and what's the actual control, and what's the genetic trigger, because something has to change genetically between these species in order to change this control of the timing. And so I think once we'll have identified what's the real mechanism, then you can try to see how this might be tweaked during evolution in order to generate these different types of metabolism.
So it sounds like you have to back up from biochemistry into evo-devo studies.
Yeah, exactly. Yeah.
Well, we certainly wish you luck with that new work, Olivier. It's been fascinating talking to you. So thanks very much for talking with us today.
Thanks. It was a pleasure.